Hey, what's going on, guys? Mr. Rouge here, and today we're talking about DNA. That is short for deoxyribonucleic acid. That's where we get those three well-known letters from. So, a brief history on DNA and how its structure was discovered. See, back in the 1950s, scientists knew that DNA was the molecule that was responsible for giving you your traits. What they didn't know, however, was how did DNA work? How did it function? In order to figure out how something works or how something functions, the first thing you should understand is its structure, its shape. What does it look like? What are the pieces to it? Okay? So in the 1950s, there was a big race amongst different scientists to discover the shape or structure of DNA. And that's what we're talking about today. Today, all we're going to talk about is the structure of DNA. Well, in the 1950s, there was this race, and there was many scientists looking for the structure, but the main ones that stuck out is one group of scientists named Watson and Crick, and then we have another scientist named Rosalind Franklin. They're both working to try to discover the structure of DNA, and Rosalind Franklin, what she did was she did what's called X-ray diffraction. She would take a molecule and put a crystal over it, and she would shoot X-rays at it, and that would produce a picture. Well, she created this picture, and this was very hard to get, but this picture right here, it's a very famous picture. This is of DNA. This is of a DNA molecule. And Rosalind Franklin, she actually met with Watson and Crick. They tried to collaborate for a short time, and one of the gentlemen saw this picture, and he kept quiet, and he said, I know what this means. He took that information back to his partner, and they worked using this information they saw from the picture, and they found that DNA is a double helix. What that means is it's like a twisted ladder. There's two strands to it, and it's twisted around. Okay. Later, they published their work. Watson and Crick was accredited for finding the structure of DNA, and Rosalind Franklin sadly passed away. Since she worked with X-rays, she got cancer and she passed away, and she did not get credited for finding DNA structure until it was brought out that they had actually seen this picture from her, and that's what helped lead them to that discovery. Okay, but now we do recognize her as a big contributor in discovering DNA structure. Okay, so these are the notes for DNA that I want you guys to take down, and then I'm going to show you guys a little bit how it all works together. Okay, so these are the notes I want you to take. First of all, DNA stands for deoxyribonucleic acid, DNA. Okay. DNA shape, like I had discussed, is a double helix. Again, that is a twisted ladder or staircase shape. So take a ladder, twist it around, and that looks like DNA. A little remind from semester 1, DNA is a polymer. It is a large molecule. That means it's made up of smaller molecules. And the repeating monomer, its building block, are nucleic acids. I'm sorry. Let me say that one more time. Are nucleotides. Okay. DNA is a nucleic acid made up of nucleotides. That's the building block. Each nucleotide, each building block of DNA consists of three parts: a phosphate, a sugar. That sugar is deoxyribose. Remember, anything that ends in ose is a sugar. Okay, so specifically, what type of sugar is found in DNA? Deoxyribose. And lastly, a nitrogenous base is also in DNA. There are 4 different types of bases. So in all of the nucleotides, the phosphate and sugar is going to stay the same. In all of the different nucleotides, phosphates and sugars are the same. However, the nitrogenous base may be different. And there's 4 different types of nitrogenous bases that may be found in a nucleotide. Okay? The 4 different types of bases are adenine and thymine. And we say that they are complementary of each other. What does that mean? Does that mean they say nice things to each other? No. Complementary means that they fit together. They're matching. Okay? The other two bases are cytosine and guanine, and cytosine and guanine are also complementary of each other. They complete each other. They fit together, okay? And I want you guys to write this down. Adenine and thymine are complementary. Cytosine and guanine are also complementary. Now, if I say, what about adenine and guanine? What about these two? They are not complementary. They do not fit together, and you'll see that in just a moment, okay? We also say that the two strands of DNA are anti-parallel. What does that mean? Well, the two strands of DNA run in opposite directions, okay? So, for example, I had said that DNA is like a twisted ladder. Well, let's say if we untwist it and straighten it out, and we look at the two strands of DNA, we would see that one strand is facing in one direction, the other strand is facing the opposite direction. This is anti-parallel. So this is what this is talking about. Okay? Anti-parallel, each strand is running in opposite directions. Okay? Let's go and clean that up a little bit. The backbone, okay? The backbone of DNA. If I refer to the backbone, what I'm talking about are these side rails, okay? The backbone of DNA is made up of the phosphates and sugars. The rungs, or the steps of the ladder, okay, these middle sections, those are the nitrogenous bases. This is where I'll find the adenine, thymine, guanine, and cytosine, okay? Do me a favor. Go ahead and take note of everything on this slide and give that picture a quick sketch, please and thank you. Clean it up one more time. And also be sure to give a little sketch of DNA shape, that double helix, okay? Doesn't have to be too fancy. Perfect. All right, let's go ahead and move on, and let's see how all this fits together. This is the double helix, the twisted staircase. That is DNA shape, okay? Now, let's take a look at the nucleotides. I remember I said there's three parts to a nucleotide. We have our phosphate, we have our sugar. What kind of sugar is that? You got it. Deoxyribose, that's the sugar found in DNA. And lastly, we have our nitrogenous base. All three of those pieces make one nucleotide monomer, okay? So DNA is the polymer, and the monomer is nucleotides. Give that a quick sketch as well, please, okay? Okay, so let's see, what did I mean about those complementary bases? So we have adenine and thymine, those are complementary of each other. Guanine and cytosine, those are complementary of each other. And if you stop and just analyze their shapes, you'll see that adenine and thymine do in fact fit together. And guanine and cytosine do in fact fit together. Okay? So that's what I meant by complementary. Adenine cannot fit with guanine or cytosine. Thymine can't fit with cytosine or guanine, and so on and so forth. They can only fit with each other. Okay, adenine, thymine, cytosine, guanine. An easy way to remember this is, where do you find apples? You find apples in trees. So A pairs with T. A pairs with T. Where do you park cars? Well, cars go in garages. So C pairs with G. Apples in trees, cars in garages. That should help you remember, okay? All right. So let's see how the monomers actually build DNA. So we have our monomers. This is only making one side of DNA, one strand. Remember, DNA is double-stranded, so this is one side. We need to make the other side. Check this out. In order for this nucleotide to fit over here, look what it has to do. It has to flip around. Okay, and now we're building that double strand. And we could see that the phosphates and sugars are making the backbone, okay, the side rails of that ladder. Phosphates and sugars make the backbone of both sides, okay? So that's the same over here. You got a phosphate, and then you got a sugar. The rungs, or the middle portion, are made of the nitrogenous bases. And again, those nitrogenous bases can be any of the four types: adenine, thymine, guanine, or cytosine. Okay? And if we look at this, we can see that one side of the ladder faces one direction, the opposite side faces the other direction. We call that anti-parallel, okay? That's what anti-parallel means. They're not perfectly parallel, but they're flipped. All right, folks, that is it for the structure of DNA, and I'll catch you all next time. Peace.